Thank you for the introduction. Hello, everyone. My name is Xinhai Pan, and I'm a fourth-year PhD student from Dr. Shouwei Zhang's lab. And I'm very happy to be here to present my recent work called TemSOMap, which is a computational tool to map lineage-resolved single-cell RNA-seq data with spatial transcriptomics data. So spatial transcriptomics data, as we are already very familiar, is able to visualize gene expression in the context of a tissue. However, depending on the technology we're using, a lot of the spatial transcriptomics data suffer from the limitation of either gene throughput or the resolution of the space. So what people come up with the solution to introduce additional single-cell RNA-seq data to either impute the missing genes or deconvolute the cell types of the ST data. So here, reversely, if we treat the spatial transcriptomics data as a reference, and what we can do is to infer the spatial coordinates of single cells. So this problem is like different from the deconvolution in the fact that you are not just predicting the cell type percentages, but you're actually trying to infer the spatial coordinates on a finer single cell level. Therefore, we might need additional information from the single cells to perform this task, such as the temporal information. So in order to obtain temporal information for the single cells, we refer to lineage tracing technologies. Recently developed CRISPR-Cas9-based lineage tracing technologies has allowed us to sequence the lineage barcodes and single-cell RNA-seq data at the same time. So briefly talk about how this experiment works. So it usually starts with one cell at the root, which is usually an embryo or a stem cell. And we inject this root cell with a blank lineage barcode, which is an exogenous sequence. And throughout the generations of cell divisions, this lineage barcode can mutate, which corresponds to insertions or deletions on the sequence. So in the end, what we get is the cell at the leaves, which contains the mutated lineage barcode, which can be served as a dissimilarity measure and can be used to infer the hidden lineage tree. So as I mentioned, this lineage barcode can be simultaneously read out with the cell by gene matrix we have. So it's paired dataset. So with those two types of dataset we have access to, we would like to ask a very general question. It's like how to jointly model the spatial temporal development of cells. So this is a very difficult question to answer at a glance. And I would like to use some example to demonstrate our assumption. So we look at a very old experiment, which is on mammalian prefrontal cortex. So we are this experiment labels the neuronal stem cells at the inner side of the cortex and observe the position and observe the origin and the final position of the neurons at different developmental stages. We can observe that this stem cells starting from the inner part will start at some development stage, will start to divide asymmetrically and produce functional neurons that will further migrate to different layers of the cortex. In the end, what we can observe nowadays from the spatial transcriptomics data is this layered structure of the mouse cortex. So the panel on the left also tells us that the cells from the same origin of stem cells tends to have similar migration pattern and end up in a closer spatial proximity. Therefore, we think that by the integration of the lineage-resolved single-cell RNA-seq data and spatial transcriptomics, we are able to study the spatial temporal development of cells. With those motivations, we developed TemSOMap. So TemSOMap takes two inputs. One is the paired single-cell RNA-seq data with lineage barcode. We have two matrices. It's a cell by gene matrix on the top and a cell by lineage barcode matrix at the bottom. The other input is the spatial transcriptomics data, which can be considered as a spot by gene matrix. And for each spot, we have their spatial location information. We developed the loss function. The first one is the expression loss that compare the gene expression from the two inputs. And we also implemented a lineage loss that compared the lineage information from the single cell data and the spatial location information from the spatial transcriptomics. Using a gradient-based optimization program, we are ending up with the output of the TemSOMap, which is the cell mapping matrix M, which is a cell by spot matrix that maps single cells onto the spots. And using this matrix M, we are able to reconstruct the spatially resolved cell lineage plus transcriptome map. So with that, I would like to discuss a little bit more detail about the processing of the cell by barcode matrix, which might be less familiar with the audience. This cell by barcode matrix will first need to be transformed into a cell lineage tree. This task is usually done by a lot of heuristic-based methods from phylogenetic tree inference, such as neighbor joining. And this tree can encode both pairwise lineage distance between the cells, and clonal clustering information. So the clonal clustering information referring to cutting the tree at a certain edge, and then the descendants for each leaf node at this certain depth are considered coming from the same clone. So moving on to some mathematics of TemSOMap. The foundation of TemSOMap is to align gene expression of cells with spots. We use a linear decomposition way to model this. So consider a spot by gene matrix Y. We can easily model it by the product of a spot by cell matrix M and the cell by gene matrix X. So here, the cell mapping matrix serves as a transition probability matrix from cell to spot. Therefore, we enforces probability constraint. So each column sum of M is 1, and all the entries of M are non-negative. And this cell mapping matrix M is the objective and what we want to learn from the model. To compare the inferred spatial transcriptomics data, which is the spot by gene level data, and can be computed as M transpose X, with the input reference spatial transcriptomics data Y, we use cosine similarity loss function. So this idea is motivated by the state-of-the-art method called tangram. This cosine similarity loss is able to capture the relative distribution of the gene counts, and we apply this to both spot and gene, so it captures the distribution across both categories. Additionally, we have some other constraints. The first one is a density constraint. So, which here the vector D corresponds to for certain spatial transcriptomics data, we might be able to infer the cell density at each spot. With our M matrix, we are also able to model the density by calculating the row sums of the M matrix. And we can use a KL divergence term to make the density distribution between the two as similar. Further, as I mentioned, we are trying to infer the cell locations, the locations of the single cells. So we want the M matrix to be sparse. So we incorporated an additional entropy constraint. So moving on to the lineage part, in order to compare lineage with space, we will first need to infer the spatial coordinates of single cells. And this term can be calculated in a similar manner by the fact that the cell spatial coordinates can be represented as the spot spatial coordinates multiplied by the spot by cell matrix M. And with the inferred cell spatial locations, we are able to calculate the pairwise distance D using Euclidean distance. And the pairwise distance matrix D are then compared with the inferred lineage distance on the lineage tree. So we normalize the two distance matrices, and we use a MIC loss to calculate the dissimilarity between those two. Additionally, as I mentioned before, the lineage tree not only encodes the distances between the leaf nodes, it also has more global patterns by representing the cells in a clonal pattern. So let's say this is an output from a neighbor joining tree, which can be unrooted. So at all those 5 red axes, you can treat them as a root node, which will result in different shape of the tree topology. In order to obtain better clonal pattern, we are gonna perform a reroute and a rebalance of the tree to find the best clonal markers. In the end, we have this 1D vector that represents the clonal IDs of the single cells. So this is kind of like a clustering of the cells. And we incorporate a clonal pattern loss by calculating the sum of the intro clone distances. And we try to minimize this too. So with all the terms we've introduced, we end up with a weighted total loss, and this loss can be minimized via Adam optimizer. And in the end, we are able to obtain the cell mapping matrix M. So with that, I would like to move on to results. As the previous presenter also said, when we think about validation, we try to find some ground truth and we refer to simulations. But simulating spatial data is very difficult, in order to generate ground truth for paired gene expression, lineage barcode, and spatial coordinate data, I developed another method called spot testing. So I will briefly introduce how spot testing works. So spot testing with a model called cell state tree, which basically characterizes the developmental trajectory between cell types. We can run a Brownian motion model to get the gene expression differences between those cell types. And then we have another additional input, which characterizes the actual single cell relationship, which is the cell division tree. We run asymmetric divisions on this cell division tree, and we're able to infer the cell states of each individual cells. Then we run another Brownian motion model with some post-processing, and we are able to get a single cell RNA-seq data. And at the same time, we can introduce random insertions and deletions while iterating on the cell division tree, so we can get paired lineage data and single cell RNA-seq data. So from this lineage tree, we want to further model the spatial data. And I developed the cellular automata model to try to embed the cell division tree and the cell asymmetric division and migration events in 2D. So in this way, we are able to get a spatial map at a single cell resolution. To get spot level data, we can perform tessellation and binning the expression in each voxel. In order to validate TemSOMap, we are gonna treat the textulated spot level data as a reference ST data, and the paired single cell RNA-seq data and lineage barcode as the query data. So with the simulation of spot testing, we are able to label cells based on two categories. One is the cell type relations, another is the clonal relations by cutting the cell division tree at the early depth. With the ground truth, we are able to visualize how these cells distributed in space, both in terms of cell types and clonal IDs. Using TemSOMap on the lineage data, we're able to infer the single cell coordinates and compare it with the ground truth of the single cell spatial coordinates. We can see from the observation that TemSOMap is able to recover the spatial pattern in terms of this example, more about the spatial ID, clonal ID clustering. So in spot testing, we have this hyperparameter called migration rate, which controls how frequently cell migration will happen during simulation. And tuning this parameter, we can control how much the 2D spatial clustering pattern are controlled by either cell types or clonal IDs. To be more explicit, higher migration rate corresponds to more cell migration happening, which are guided by the cell type. So cell types behave, same cell types might behave similarly in terms of migration, which end up in clustering of the cell types. If fewer and lower the migration rate and fewer the cell migration event happening, the spatial map will show more spatial pattern in terms of clonal ID, as shown for this example. For this comparison, we compared with the state of the art, which is Tangram, that uses only gene expression. We can show that with varying migration rate, TemSOMap is able to more accurately infer the cell locations calculated by the MSE loss between the single cell coordinates of inferred results and the ground truth. So moving on, some real dataset experiment, and in terms of real dataset, this is very difficult because it's very hard, and it's very hard to map two separate ST data and the lineage trace the single cell RNA-seq data, and there's no ground truth. But what we find is two separate datasets. On the left-hand side, we have this stereo-seq dataset, which is a spot level dataset. that sequence mouse embryo at E9.5 stage. And then on the right-hand side is also a published lineage trace RNA-seq dataset that also sequenced the mouse embryo at the same stage. By applying TemSOMap on this dataset, we are able to reconstruct the spatial map at a finer resolution than the reference spatial transcriptomics data. And if we take a deeper look at specific marker genes that might correspond to different spatial activities, for example, this PAX6 here have been associated with eye development and neuronal cells, and this GATA5 here can correspond to cardiac muscle, smooth muscle diversity. We can see from these three panels, the first column is the input spot level spatial data. The second one is the inferred single cell level data. And the third one is the inferred spot level data, which is simply calculating M transposed times X. So we can compare the first and third to show that the TemSOMap inferred results can have very similar spatial clustering pattern as the reference. And at the same time, we are able to infer the spatial distribution of cells at the single cell level. And more excitingly, because we have this paired lineage information of the cells and we map them onto space, now we can perform analysis in the spatial temporal manner. What we do here is that we can start with a subtree on the cell lineage, which is this subtree A, which comprises 704 cells with diverse cell types, which indicates at this current depth of the tree, the cells are multipotent. And we can go down the lineage tree and find the two separate paths. One is the subtree B and C, and another is subtree D and E. And surprisingly, these two paths end up in two separate different cell fates. The top one ends up with a majority of brain cells, and the latter one is more about AGM cells. And from using the spatial map, we can also see how the spatial distribution of the cells changes along the path. So this is like the results that validates that using TemSOMap to map the two datasets, we're able to analyze different cell phase specifications from one multipotent cell lineage. With that, I would like to summarize TemSOMap. So TemSOMap is a computational tool that maps paired lineage plus single-cell RNA-seq data onto a spatial transcriptomics data. And the additional lineage information help us to refine the cell location inference. And at the same time, we still maintain accurate spatial expression patterns in terms of both cell types and marker genes. And I want to mention that the lineage information of cells can be obtained from multiple sources. And the one that I use is like from the CRISPR-Cas9 induced genetic scars. And there are also technologies developing to identify cell lineage from other biological traits like somatic mutations of mitochondrial genes. So with that, I would like to acknowledge the co-author and my advisor, Shouwei Zhang, and all the lab members that helped me with this project. And thank you guys for listening, and happy to take any questions.
Maybe I start. So a very interesting talk. Thank you.
Thank you.
I was wondering, one of your biggest evaluation was based on simulated data, where you're jointly simulating single-cell RNA-seq, spatial data, and barcoding data. And even when you consider just the simulation of single-cell data, that's a very rich area with like a lot of methods trying to very carefully validate. So my question is, how did you validate the result of your simulation to begin with before you start validating the method?
Thank you. That's a very good question. So I think in terms of simulation of the single-cell RNA-seq data, most commonly used are more statistical based methods that try to basically generate data points using certain distributions. But my philosophy for simulation is more about trying to use like more like computational, in a computational manner, trying to model the biological events that actually happens, which can have better performance in terms of integrating different data modalities. When you think about how to sequence in this example, when you think about how to sequence the cell type changes on the cell division tree, you will have to come up with a model that model the changes along the tree, and at the same time, you will need to come up with a model that model the differences between the cell types. So I think simulation, yeah, it's very hard to validate how good the simulator is, but in terms of the comparison with other methods, I think first is like we can compare the methods on a biological setting, and also like with different hyperparameters, we can change the correlation between modalities and try to have a better understanding of the big picture.
If your simulated data doesn't look like real data,
oh yeah.
Hard to, right? So that's why the statistics.
Yeah, in terms of that, in Spatial we are also able to fit a real data set, and we can show that Spatial simulated data can have similar cell type percentages, spatial distributions, and also like just like spatial clustering pattern of genes. But like we're not showing here, but how we achieve that is by also fitting the parameters that we give Spatial using a reference real data set. In that way, we can generate data that more resemble the real data set. But in this case, we are using a more like a free way, like a more synthetic one.
I have a small question. So I know that this migration would also take into account the cell division
yes.
and stuff like that. So there is a new protocol called lineage-resolved, which takes the slices at different points. So one can think about like modeling migration. So have you looked into that?
I be honest, I haven't looked into that specifically, but it would be very, yeah, I think that's a very good idea to kind of look at different slices and try to investigate how cell migrate. And I still think right now it's very difficult to kind of just hypothetically think of how cell migration happens, but we definitely need to learn more from the data. We need to have more paired data across slices, across different time points, and in the 3D manner as well, in order to have better understanding of migration.